Hello everyone. So in the last session we were studying about the comparison between genes and chromosomes. So in this session we are going to study about the linkage and recombination. Before starting the concept, I would like to tell you about one scientist known as T.H. Morgan. Okay, so T.H. Morgan, Thomas Hunt Morgan. Who is he? He is the father of genetics. Genetics means it is a study of genes, or it is a branch of biology which deals with the study of genes. So T.H. Morgan, what his contribution, what he did, what's his discovery, so all those things we'll study first. Later we'll move on to the linkage and recombination. The Thomas Hunt Morgan, and with his colleagues, he worked by taking one organism known as fruit flies. Those fruit flies, scientific name is Drosophila melanogaster. Drosophila melanogaster, scientific name for fruit fly. The fruit flies, it will be hovering on the fruits. So what is the purpose of taking or selecting the fruit flies? What is so special in that fruit fly? We will find out the reasons behind it. The fruit fly is, so these are easily grown on the synthetic medium. Okay, so this is the first reason for which he selected the fruit fly. Not only this one, we have few more reasons for selecting the Drosophila melanogaster. Drosophila melanogaster, it's a fly. It will be in this size. And those fruit flies are easily grown in the laboratory condition on the medium, on synthetic medium, or by using some sugar or some like that kind of medium, we can grow the fruit flies. And the second one is their life cycle ends in 1 to 2 weeks. Their life cycle ends. It means the egg stage and then larva and then pupa, it completes its life cycle within 2 weeks. Okay, so in the short term he can produce or he can grow number of fruit flies. And in single mating of male and female fruit flies, it can produce number of progeny. So that is also one reason behind it, why he selected fruit fly. In single mating of male and female fruit flies, the female fruit fly can produce number of progeny, and male and female Drosophila or fruit fly can be easily distinguishable. So this is also one reason. So by seeing the fruit fly, by measuring the size or by seeing the size of the fruit fly, we can easily tell which is male Drosophila and which is female Drosophila. And the fifth one is it has number of hereditary variants. It has number of hereditary variants in sense so many Drosophilas are there which have yellow body, and few of them will be having brown body, few of them will be having white eye, and few of them will be having red eye. So these are the number of variations seen in fruit flies. In sense, what we studied in the introduction part, we studied about the pea plant. So in the pea plant, the Mendel discovered number of contrasting character. He took the 7 contrasting character for his work. Likewise, as we studied in the pea plant, as it was having the 7 contrasting character, this is also having number of hereditary variations. In a male and female cross, it can produce the various hereditary offsprings. So we'll see that later. So with this, we'll move on to the linkage and recombination. So what is the purpose of studying the Drosophila melanogaster before linkage and recombination? Here I'll tell you the linkage and recombination. I didn't told you about his discovery. He worked on the Drosophila melanogaster, and what's his discovery? What he found out? Following the synthesis of chromosomal theory of inheritance. We studied about the chromosomal theory of inheritance in the last session, right? So what it tells, the chromosomal theory of inheritance, the chromosomes, it can itself assort independently. The chromosomes, number of genes are present, so and so we studied in the last session, right? So, but with the following those, principles, and with the following those knowledge, Morgan, he discovered the basis for the variation occurs in sexual reproduction in offsprings. So based on the sexual reproduction, whatever we are having the offsprings which get reproduced by means of sexual, they'll be having some kind of variations. So because for what is the reason behind it? Why the offsprings which get reproduced by means of sexual reproduction, they're having variation? Why so? So something is there, something we are missing, something we didn't learn about the variation. So that variation, that concept we are studying in the linkage and recombination, that was clear-cut described by T.H. Morgan, but he didn't, he cannot work on human. Okay, so because of the reason, he took the fruit fly. Okay, he chooses the fruit fly. So we'll move on to the linkage and recombination. So linkage, it literally means something is linking. What is that something? Something is nothing but genes. Genes are linked with one another. So that study we are describing in the linkage. So consider this is one chromosome. Okay. One more chromosome we have. They both are non-parental. In the meiosis cell division, consider this as one paternal chromosome and maternal chromosome. Okay, so at the time, at the stage of Pakiti, what will happen? The crossing over occur. the crossing over or recombination. What is the purpose of recombination? The recombination purpose is it has to share the genes of non-parents. Okay, so this chromosome it has to share its genes, and this chromosome it has to share its genes. So that phenomena is going to occur in the packaging stage. The recombination, what I told you, the recombination occurs in the packaging. Okay, so just keep that in mind. Now we are in the linkage concept. Consider here we are having a gene. I have named this gene as capital A. Okay, somewhere here I am drawing, I am pointing out one more gene and I am labeling this as B. On one more parent I am drawing one gene, naming that as D. Here C. Okay. Now crossing over occurs. After the crossing over, so how the crossing overs occur? So in this diagram I am going to show you. So here what you have to observe here the genes are far away. They are not linked with one another. Linking means, in the next example I will tell you. So these genes are far away. They are very in the extreme ends. So this gene is here, one more chromosome. This gene will be there on this limb. This gene is there. Okay, so crossing over means the chromosomes are crossed with one another. What will happen in the next step? The genes remain constant. Here in this part gets separated. The non-parental chromosome shared its genes with and this part. These two parts get shared in the non-parental chromosome. So what I should write here? Here I should the C gene, and here one more chromosome I have to draw, D, and here it comes the B. So because of this phenomena, the genes get shared in the non-parental chromosome. When this chromosome exerts itself in the gamete, when those gamete reproduces or fertilizes, after the fertilization, whatever the offsprings are going to form, those offsprings will be having the both parent and male and female, or father and mother's characters. Because of this reason, what will happen if the genes are closely packed? So one gene I am drawing here, one more gene. Consider these two genes are packed, tightly packed. Okay, so one more chromosome I should draw here, naming that as D and C. Alright, so what you have to draw in the next diagram? So after the crossing over, after this crossing over, the C gene will come in this chromosome and it remains empty. Here the genes did not travel or transmit to this non-parental chromosome. So these are the things, or these are the mechanism going to form in the linkage. However, the genes are very far. If the distance is more in between these two genes, the recombination the recombination level will be high. If the genes are tightly packed, the chances of recombination is very less. Okay, so here it have not shared its gene with the non-parental chromosome. So recombination chances of recombination is less. The separation of genes are also less because of the what is that? Extreme ends, presence of these genes in at the extreme ends, the recombination occur in this possibility or in this example. So that's all about linkage. Definition is physical association of two genes. Okay. So with this we'll take one example. What example we are going to take here? We are taking the Drosophila melanogaster to explain about the linkage. Here, Drosophila melanogaster, we are taking two characters of Drosophila melanogaster, like one Drosophila, female Drosophila, what we are choosing, it is having yellow body and white eye, okay, which is a female one. When we cross this female Drosophila with the brown body and red eye, which is a male one. Okay, so we have two characters, yellow body, white eye, brown body, red eye. Consider brown body and red eye, it is in a dominant position or dominant, this is the dominant one. Okay, and this is the recessive one. According to Mendel's principle, when we take the two characters, what we should in the F2 generation, what ratio we have to get? We have to get the 9:3:3:1 ratio. Okay, but the result is far differ from the Mendelian principle. But in the F2 generation, we won't get this ratio. We'll find out how. So we have a structure here. So look at the board. I have drawn number of boxes, number of lines. I guess you can't think or you can't recognize what are these. Let me tell you, one by one we'll go. Okay, consider these are the genes. Okay, which is located or which is present very close to one another, and consider this as one X chromosome and one more X chromosome. Because we have taken this as female, we have to show XX type of chromosome. Why? This is sexually linked inheritance. On that, in this side, I have drawn this strand, which means this is the X chromosome, and little bit limb I have drawn here. Now hook type. This is considered as, or that is considered as Y chromosome. When we cross these two parents or these two male and female Drosophila, what we got in the F1 generation, we got one more thing I should tell here. So yellow body character or that character represent by this gene, okay, white eye represent this gene, and in this side we have brown body and red eye, right, which is dominant. As I told you, the dominant character represented by the plus sign. Okay, so Y plus and W plus in sense it is brown body and red eye. We'll cross these two. How to cross these? this X chromosome will go with this X chromosome. Here we are having Y plus and W plus. Here we are having only Y and W, Y and W. This one X chromosome will go with this X chromosome. In the F1 generation, what we got? One X chromosome will be as it is, and one X chromosome will be having a dominant gene Y plus and W plus, and this X chromosome will go with the Y chromosome. That's what I have written here. So phenotype, in the phenotype, what we have to write here? The Y plus is dominant over this Y, only Y. Wherever the Y plus sign came, you have to show this Drosophila is brown body. Wherever W plus sign comes, you have to show that as red-eyed Drosophila. In the combination of these two, we can write a wild type. Okay, all the wild Drosophilas will be having brown body and red eye. In this side, what we have written? We have written one X chromosome and Y chromosome. So X chromosome, on the X chromosome, these two genes are present. Yellow bodied, white eyed. Yellow bodied, white eyed. Though the Y chromosome doesn't carry any gene, we can't show any phenotype wherever the Y chromosome is present. One more side, we have written like this. So if the box is empty, only for this, only for your convenience I have drawn here. If the box is empty, consider that is nothing but dominant gene. Here nothing, I have not shaded this. It means these two, out of these two genes, this chromosome having these two genes, it is dominant. Because of that, we have to write the phenotype as wild. And in this side, both the chromosome carry the recessive gene, so yellow and white. So 98.7% of the offsprings in the F2 generation. So what is this? I didn't told you. We have crossed these two offsprings. What we got in the F1 generation? No, we crossed or we intercrossed these two offsprings. In the result, in the F2 generation, we got the number of offsprings. Still 4 more are there, but this is more than enough. In the F2 generation, 98.7% of offsprings resemble the parent. Only 1.3% of offsprings will be recombinant type. So that frequency, what we are showing in the percentage, right? So the frequency of recombination. So the frequency of recombination is more. Like the parent resembling offsprings are more, the new combination or the new chromosomes are less. Why? Because the genes are linked. The genes are physically associated with one another. What will happen if the genes are located far away? Okay, one more example I should show that. So we have one more example here. So, but in this example, I have shown the genes are located far away from one another. Okay, W means white-eyed. The chromosome, the gene's phenotype is white-eyed. M means miniature wing. The Drosophila will be having wings, right? So those wings will be miniature, the size will be small. So those two character Drosophila, female Drosophila, when we intercrossed with the wild type, okay, W plus means the red-eyed and M plus means normal wing. When we intercrossed these two, we got in the F1 generation, the female become the wild type because it crossed or it takes the W plus sign gene. It means red-eyed and normal wing genes it takes. So because of that, this Drosophila become a wild type. But here this X chromosome goes with the Y chromosome. The male become white-eyed and having miniature wing. When in the F2 generation, when we intercross these two Drosophilas, if we cross this female with the male, what we got? The 68% of the offsprings, only 68% of the offsprings resemble the parent. The 32% of the offsprings became recombinants. Okay, why? In the first example, only 1.3% of the offsprings will become recombinants. The leftover, the rest of them, 98% resembles the parents. But in this cross, we got 32% of the offsprings. The 32% are of recombinants or new types. Why it is so? Because the genes are very far from one another. They are not linked with one another. The recombination chances are more. Right. So that is why they have given this example. Learn it and don't forget the scientist's name, Thomas Hunt Morgan, and then for what purpose he chooses the fruit fly, what is the scientific name of fruit fly. All those things are important. And what is linkage, what is recombination, one line definition they'll ask you. Learn it. Thank you.